In this analysis, we will be determining the amount of chloride in a solution based upon its complexation with silver. It will form a silver chloride precipitate, which will cause a color change due to Fajan's reaction. In this titration, it is crucial to add dextrin to each flask. The dextrin can be pre-weighed, only weighing about 0.1 grams, and kept in weigh papers until ready to be added to each flask. It can be added well in advance of the titration. And no, I did not throw the weigh paper on the floor. There is in fact a garbage on the other side of the desk. After the dextrin has been added to the solution, add additional water to break it up and ensure that as much of it is dissolved as possible. You can rinse down the sides with the ionized water. The indicator used in this titration is dichlorofluorescein. The indicator can adsorb onto the surface of the silver chloride particles, but only when there is no excess chloride present in solution. When all the chloride has been precipitated by the addition of silver, the fluorescein will then adsorb onto those particles, going from a yellow color to a pink endpoint color. Your first titration will be a rough titration, where you add the fluorescein right away. You will then rapidly add titrant to reach the endpoint, or at least a rough approximation. The reason for this is that the addition of fluorescein to the titration flask is detrimental to the proper visualization of the endpoint. The fluorescein accelerates the photoreduction of the silver ions, leading to the formation of black particles. These black particles make it difficult to visualize the color change from yellow to pink. For future titrations, you will not add the fluorescein indicator until you are a couple of milliliters away from this rough endpoint. All other aspects of the titration are the same. You will add the dextrin before doing the titration, you will rinse it down with some DI water, and you will carefully record the volume on the burette before beginning your titration. Since a rough endpoint is already known for this titration, you can add titrant rapidly until you are a couple of milliliters from that endpoint volume. Do remember to swirl and occasionally rinse down the sides of your flask. It is still important to maintain proper analytical technique, even though you are going rapidly. Once you are within a few milliliters of the approximate endpoint, you will want to add the dichlorofluorescein indicator solution. You only need about 4 or 5 drops in order to be able to see the color very nicely in the solution. Now proceed to slowly and carefully add titrant until you begin noticing a pink color appear where the titrant solution reaches the flask. As with other titrations, the longer this endpoint color persists, the closer you are to the endpoint. Continue to swirl the flask and rinse down the sides periodically to ensure that all reagents are mixed fully. The color change is subtle and is not very easy to visualize in this video. However, you do want to have the solution change to a pink color, but a very light pink color, not a very dark Pepto-Bismol pink. You may also note that the previous flask used for a titration, sitting on the right of the screen, has now become more of a grayish pink color. This is because of the photoreduction of the silver in solution, as well as a settling of the particles over time. For this reason, you should not compare your color to the color of previous titrations. There is a significant change over time. Here you should be able to see that one drop turns the solution to a slightly pink color, removing most of the traces of yellow. For a better comparison, here is a still photo of the endpoint and the original color of solution, as well as an overshot titration which clearly indicates a too dark pink color.